welcome to chapter 4.2. So 4.2 is the first section where we're going to start to talk about the biomolecules. So we're going to talk specifically about lipids and sugars in this lecture. And in the next section in 4.3, we'll talk about the other two biomolecules. So before we start into 4.2, we need to talk about biological macromolecules. Okay, so biological macromolecules, so macro means big, right? Micro means it's small. So these are large biological molecules or large biomolecules, if you remember the definition from the previous lecture. So these are large molecules that make up cells and living things. Okay, all macromolecules are organic, they're organic molecules, so that means that they contain carbon-carbon bonds. Now, macromolecules are what we call polymers. So poly means many, and mer means unit. So polymer literally means many units. Okay, so macromolecules are polymers, so they're molecules that are made of many smaller units. Okay, we call these small units monomers. Okay, so we can put monomers together to make polymers. So when we talk about the different classes of macromolecules, we have four that are found in living things. Okay, we have lipids, so we refer to these more as fat oils. We have carbohydrates or sugars. We're going to talk about some polysaccharides. We have nucleic acids. This is our genetic material. These are things like DNA and RNA. These are the nucleic acids. Okay, and then we have proteins. So we've used the term protein quite a bit in this class so far. Proteins make up things like enzymes, receptors on the outside of the cells. We've talked a little bit about receptors, structural components. Proteins are actually really, really important for living organisms and cells. So over the next two sections, we're going to talk about these four different classes of molecules in a little bit more detail. Okay, so in 4.2, as I said, we are going to focus on lipids and sugars. You need to be able to describe organic molecules and explain how they're formed, how we make the polymers. You need to be able to describe the structure of lipids and their key functions within the cell. So what do they do to cells? And then you need to be able to describe the structure of sugars and polysaccharides. Polysaccharide means many saccharides, saccharide means sugar, so many sugars. So polysaccharides are the polymers of the carbohydrate group. So let's start with are made up of mostly carbon and hydrogen. And lipids are important because they make up the structural components of the cell. So they mainly make up the outer membrane, so the outer boundary of our cells. Okay, they can make up other parts as well. One thing we forgot to talk about in 4.1 is the fact that compounds store energy. Okay, so energy is stored in the bonds that make up a compound. Different compounds store different amounts of energy. So different biomolecules store different amounts of energy. So some of them are higher energy storing and some are lower energy storing. So lipids, when we talk in terms of energy storage, they are some of the best energy storage. And that is because lipids are made of long chains of carbon and hydrogen. We call these hydrocarbons. Okay, so if we look here, in this image, we see that we have this long hydrocarbon chain. So we have all these carbon-carbon bonds. Each carbon has hydrogen attached to it. And because we have these long chains of carbon and hydrogen, the bonds between the carbon-carbon, the carbon-carbon bonds, this is actually one of our highest energy storing bonds that there is. In fact, this is why we think that we are carbon-based life forms, is because of the fact that carbon-carbon bonds store a lot of energy, which means that if we break this bond, it's going to release a lot of energy. Okay, so lipids are the best energy storage molecule because they have lots of bonds to store energy. We have fatty acids. So we have two types of fatty acids. We have saturated and unsaturated. So what saturated means is that we have all single bonds in our hydrocarbon chain. Okay, so that means every carbon is going to have the maximum amount of hydrogen it can have. So remember, carbon has 4 valence electrons. They're all single electrons, so they all can make a bond. So carbon can make up to 4 bonds. So if we look here, this carbon has 2 bonds to other carbons, so that's 2 of the bonds, and then it's bound to 2 hydrogens. That brings up the next 4. So this is called saturated because it has the maximum number of hydrogens we can put on it. So this carbon is a little bit different. So it has 1 carbon-carbon bond, and then it's bound to 3 hydrogens. So we still make the 4 bonds, but we're going to have 1 more hydrogen than these ones here in the middle. Okay, so that's what it means to be saturated. So unsaturated fatty acids are going to contain double bonds. So an unsaturated fatty acid has at least 1 double bond somewhere in the hydrocarbon chain. So these are unsaturated because once we make another bond with carbon, we can't put the maximum number of hydrogens on those carbons, so we're unsaturated. So unsaturated fatty acids are actually really important because these double bonds can cause what are called kinks or bends in the molecule. So this is a double bond that is causing the kink in this molecule. This double bond doesn't really change and make a kink, but this is why one of the reasons why we look at unsaturated fatty acids, because these kinks make it so these fatty acids can't pack together and make solid structures. Okay, so we find unsaturated fatty acids in a lot of oils because they're going to remain liquid because they can't pack tightly. Now, saturated fatty acids, so these are going to make up things like butter and solid fat because they don't have any kinks, so they can pack closely together and make solids. Okay, so some examples of lipids is we have fatty acids like we just talked about. We have triglycerides. This is what they look at like if you have high cholesterol and they look for heart disease, they're going to look at your triglycerides. But the most important lipid that we're going to talk about is these phospholipids. These are what make up our cell membranes. In fact, in 4.5, we're going to talk more about phospholipids because they are so important when it comes to cell biology and the structure of the cell. Okay, so lipids store a lot of energy, but they're not the molecule that we're going to break down first for energy. That's going to belong to the class of sugars, so carbohydrates. So when we're making polymers, and this goes for lipids and for carbohydrates, what we're going to do is we're going to take each of the individual monomers, so those single units, and what we're going to do is we're going to cause what is called a dehydration reaction. Okay, so the hydrogen on this glucose is going to combine with this OH group on this glucose so we can make water. So this is why it's a dehydration reaction, is because we're removing water. Okay, this is what's going to join our 2 monomers together to make a polymer. So in lipids, we have the same thing. It undergoes a dehydration reaction to make the long chains and our polymers. Okay, so the single units for carbohydrates, we call these monosaccharides. Okay, so these are our 1 unit. We have 3 monosaccharides that make up all carbohydrates. They're glucose, fructose, and galactose. So glucose is our main buddy. This is what our bodies break down to form energy. Fructose, this is found in lots of fruit sugars. Okay, and galactose, we'll talk about this one in a second. So we can combine these monosaccharides to make different disaccharides. So disaccharide means we've taken 2 monomers and made what we call a dimer, or a 2-unit carbohydrate. Okay, so our common disaccharides are lactose. This is milk sugar. So this is going to have glucose and galactose in it. We have sucrose, which is going to be glucose and fructose. This is the main component of fruit sugars. And then we have maltose, which is glucose and so glucose and glucose together, I believe. Okay, so sugars are kind of interesting. Once we start putting more monomers together past the disaccharides, we make the polysaccharides. So these are our long chains of sugars. So polysaccharide means many sugars, many units. Our main polysaccharides are starch, glycogen, and cellulose. The thing is, we need to talk about the arrangement of sugars. So we can actually make two types of sugars. We call them the D and L sugars. Okay, so whether something is a D or L sugar depends on these bonds right here and what's attached to them. So which side the OH is on. So when the OH is on this side, it's what we call a D sugar. If it's the OH is on this side, it's what we call an L sugar. Now this is important because these two types of sugars, one is man-made and one is found in nature. So D sugars are our natural sugar, and this means that our bodies have the molecular machinery to take polysaccharides like starch that's made of lots of glucose chained together, or one of our disaccharides, and it can break this bond and make it back into two monomers and then go and use those monomers to make things like energy and to help us get through the day. Okay, with L sugars, because this bond is going to be different, it's actually going to be up instead of down because of the orientation at this fifth carbon. Those L sugars can't be broken down by our bodies. In fact, though like aspartame is a L sugar, we can't break aspartame down. So if you drink a lot of diet soda, you're going to drink a lot of aspartame. So what happens is it just builds up in your bloodstream and you're going to start peeing it out. But fake sugars like aspartame, these L sugars, have been linked to memory problems because of them being able to build up. And they can cross the blood-brain barrier and build up there, and that's going to interfere with our memories. Okay, so starch is made of D sugars, glycogen, so this is the polysaccharide stored in things like meat. Starch is the polysaccharide stored in plants, so things like beans and potatoes contain starch. Your steak has glycogen, so we can break both of these down. Now, another polysaccharide is cellulose. So cellulose is the main component of plant cell walls. This is what makes them rigid and makes plants stand up. Cellulose has the opposite bond of this, so it's facing up, meaning we can't break it down. So when we eat things like celery that have lots of cellulose in it, we're actually not getting a lot of energy from it. We're getting a lot of water and we're getting a lot of fiber that helps keep our digestive tract healthy and happy, but we can't actually break it down. So things like cows and termites, they have special bacteria in their guts that help break cellulose down because they can actually break it down. We also have bacteria in our gut that can break cellulose down, and we get energy from it that way through our symbiotic relationship with the bacteria in our guts. But this is something that we couldn't break down on our own. So carbohydrates are the main source of energy for cells and for metabolism. If your body needs energy, it's going to start to burn carbohydrates first and then move to lipids. So it's still a really good energy storage molecule, but it's not as great as lipids because lipids are bigger and they can store more energy. And the amount of energy stored in the bonds for lipids is higher than that of carbohydrates, but they're still both good sources of energy. So that is 4.2. You need to know that organic molecules have one or more carbon-carbon bonds. Our biological macromolecules are things like lipids. our carbohydrates, nucleic acids, and proteins. So you need to know the difference between the structure of lipids, so those long hydrocarbon chains, and the fact that lipids are great energy storage, and that sugars are short-term energy sources, so we can use them really quickly, and that they are going to form long chains that make polysaccharides, sometimes referred to these as glycans. Okay, so lipids, long-term energy, carbohydrate, short-term energy, and we'll talk more about phospholipids in particular later in this chapter. But in section 4.3, we'll talk about the last two classes of macromolecules, the nucleic acids and the proteins, which are two of my favorites. So we will see you in 4.3.